Good afternoon, everyone. Today, I'd like to discuss with you the topic of drug repurposing for unmet medical needs, focusing on the case of anti-diabetics for Alzheimer's disease as part of a collaboration with Mass General Brigham and Imperial College in London. So this project is joint work with a PhD student in the UK, Beng, our mentor, Bella, and the US hospital's data engineer, Colin, under the senior guidance of Mark, Deborah, Sudeshna, and Joanna. Some of you may know Deborah Blacker actually from the epidemiology and geriatry department at HSBH. And this work is currently under review, so your feedback would be beneficial once we switch to the first, hopefully, and last revision. So why are we looking for other applications of anti-diabetic medications, and why focusing on Alzheimer's disease and more generally dementia? I will articulate the motivation for this work from two perspectives: global and US-centric. So life expectancy is increasing, and so is the prevalence of aging-related diseases, including neurodegenerative diseases. About 47 million people globally are affected by dementia, and this number is expected to double, if not triple, by 2050. And in the US specifically, the increase in AD cases is a major concern because it represents a huge burden for healthcare system that's already costly and ineffective. So to date, there is no approved and deployed disease-modifying therapy for AD. While pharmaceutical companies are investing in novel solutions, another idea consists in exploring retrospectively or prospectively the potential of drugs that are already approved by the Food and Drug Administration in the US or the European Medicines Agency in Europe, but for off-label use. So which existing drugs to investigate first? Would certain compounds, if any, be more effective than others at slowing the development process of Alzheimer's disease? Dementia being a multifactorial disease, it is associated with other types of illness, and among them, diabetes. Diabetic conditions manifest in multiple ways. Similar to renal failure, the impairment of cognitive functions is a complication of diabetes induced by hyperglycemia. And in 2005, the diabetic epidemic was mostly affecting southern states. However, 10 years later, the map looked quite different, as in most states, 10% or more of the adult population had a coded diagnosis of diabetes, according to a CDC surveillance report. And these estimates do not distinguish type 2 from type 1 diabetes, but the former accounts for 90 to 95% of all cases. So if anything, this map is a lower bound of the underlying age-adjusted disease prevalence in the country, putting aside the question of racial heterogeneity across US counties. And the diabetic population is at increased risk of dementia. 20% or more of older adults with type 2 diabetes could suffer from mild cognitive impairment, MCI, which can aggravate and lead to a form of dementia. And patients with MCI actually develop Alzheimer's disease at an annual incidence of between 6 and 25%, is 0.24% in the general population of the same age. So overall, type 2 diabetes is associated with a 1.5 to 2.5-fold increase in the risk of dementia. And among the known mechanisms that intervene in both diseases, one can think of inflammation, oxidative stress, and mitochondrial dysfunction. And these three factors are known to aggravate brain insulin resistance and amyloid beta accumulation in brain lesions. And prolonged exposure to hyperglycemia and hyperinsulinemia, as well as high levels of the amyloid beta protein in the brain, can further lead to deterioration of neuronal structure and function, which results in poor cognitive performance. Thus, there are valid reasons to first focus on anti-diabetics as a first class of drugs for Alzheimer's disease drug repurposing. But there are many kinds of such medications. So which anti-diabetic medications to consider? Certainly, the American Diabetes Association, the ADA, has defined a hierarchy, which includes first and second line monotherapy recommendations. But as you can imagine, these recommendations, these guidelines changed over time. In this study, we looked at two historically first-line anti-diabetics, metformin and the sulfonylurea drug class. And I say historically because nowadays sulfonylureas are generally recommended as a second-line treatment. So comparing them is interesting and relevant because they do not have the same mechanism of action. The main difference being that with metformin therapy, insulin secretion remains unchanged. Indeed, it acts by decreasing hepatic glucose production and intestinal absorption of glucose, while sulfonylurea's mechanism is based on the stimulation of insulin secretion by pancreatic beta cells, resulting then in lowered blood glucose. And another aspect to keep in mind is that sulfonylureas have been widely used for the treatment of type 2 diabetes for more than 50 years, while metformin's usage is more recent, at least in the US. The lower cost of sulfonylureas and the ease of taking one instead of 3 pills a day might explain physician prescription preferences for sulfonylureas over metformin, either overall or in certain sociodemographic subgroups. That's something we can come back to at the end, and that I would be interested in studying further. So unlike sulfonylureas, metformin doesn't cause hyperinsulinemia, which, as we mentioned before, can lead to neurodegeneration. And this suggests that metformin might have some benefits. Along these lines, there has been some recent evidence that metformin could slow biological aging. And because age is exponentially related to the onset of dementia, it has been hypothesized that metformin would reduce its risk. But the history of that drug is a bit chaotic, and this is certainly an important aspect to consider when designing an observational study. In 1995, the FDA finally approved metformin for patients with type 2 diabetes, but with prescribing restrictions. And in 2015, after many safety trials, the agency endorsed metformin safety in patients with mild to moderate kidney function impairment. But conversely, in Europe, metformin had been used for more than 50 years. So keeping these considerations, and let's now briefly describe the study objective and the underlying data sets. So the elements mentioned before encourage us to test the hypothesis that metformin reduces the risk of dementia onset in diabetic patients. And for this, we have emulated an in silico or synthetic drug trial that compares metformin to the other drug class in 2 data sets, one from the US and the other from the UK. And the rationale for this was that the replication of results across 2 data sets from vastly different settings, including differences in medical practice patterns, in data collection, in missingness, and in known sources of bias, would contribute to the generalizability of our results. So in the US, the dataset came from the Mass General Brigham medical record system, which covers a total of 8 health centers in 3 states in New England, including 2 large tertiary care hospitals in Boston, MGH and Brigham. And patients could also come from outside this area, actually. For example, Pennsylvania and Florida residents receiving care out of state. That's something common that we've observed. And in the UK, the data set came from 13 regions and more than 700 clinical practices across the country. So this is certainly more representative of a nationwide primary care network. So here are our research questions. One, what is the causal effect of metformin on dementia onset in type 2 diabetic patients? In parallel, can we estimate the causal effect of metformin on longevity? Are these effects constant over time? And finally, are the results robust to the choice of statistical estimation procedure? So these questions could be formulated in the general population as well, but here we didn't try to generalize the EHR analysis-based results beyond the case of type 2 diabetic patients. However, in addition to estimating the causal effects of metformin on end-of-life outcomes, in this patient subgroup, the team conducted in vitro systems pharmacology drug evaluation. And the goal there was to identify candidate biomarkers in the central nervous system that could serve in clinical trials targeting a broader population than the ones studied in this work, including cognitively healthy diabetic and non-diabetic patients. So let me describe the approach we take. We emulated a target trial in 2 cohorts, the US RPDR and the UK CPRD. And here is a visual representation of the emulated trial in the US RPDR. And this is very similar in the UK. So in an intention to treat analysis, we followed patients with type 2 diabetes, age 50 and more, from their first anti-diabetic drug exposure at time T0, here on the graph, onwards. And patients initiating an anti-diabetic monotherapy were selected, while those starting on polytherapy were excluded. And although the Vanderbilt criterion for type 2 diabetes includes medication prescription and diagnosis code, or HbA1c test result, we relaxed that condition by solely requiring anti-diabetic drug exposure. Given the focus of our study, that seemed, reasonable to relax that condition. And to ensure sufficient drug exposure before measuring outcomes, we required what we call a 1-year run-in period here. And one of the advantages of observational studies as compared to randomized clinical trials is the potential to more easily investigate end-of-life outcomes, such as dementia onset. Indeed, the duration of follow-up in emulated trials is often much longer. In our case, that was up to 12 years in the US and up to 16 years in the UK cohort. Here, we considered new prescriptions from 2007 onwards. One, we did so because guidelines from the American Diabetes Association changed in 2005-2006. And we also did that because Mass General Brigham switched to an electronic system in 2007, leading to better quality data. And of course, the large adoption of EHR in the US made the study possible. So yeah, that's something important to consider here. So in emulated trials, one has to be super careful with reconstructing and applying the right set of inclusion-exclusion criteria. And this is definitely something that took time for us to do and to do it right. So first, we excluded patients with chronic kidney disease, CKD, at baseline, given that this is a contraindication for metformin, but not for the other drug class, sulfonylurea. Second, to ensure that patients were incident diabetics, we required at least 1 visit with no diagnosis or prescription in the 18 months that precede the date at which the patient enters the emulated trial. Third, we required patients to have their primary care physician, their PCP, within the Mass General Brigham ecosystem. And that was to maximize the extent to which their medical history would be recorded in order to reduce the likelihood of data missingness. So there were 2 outcomes of interest, 2 times 2 events that we captured here. One was dementia onset, and the other was death without dementia. And here, dementia incidence was defined by the presence of either an ICD code or by the initiation of an Alzheimer's disease drug. And the observational study design was very similar in the UK cohort, where we considered new prescriptions from 2001 onwards instead of 2007 onwards. So here is a brief overview of our methods, and we can deep dive into them later. So we estimated the effect of metformin on dementia with death as a competing event that precludes the development of dementia. And for this, we used inverse propensity score of treatment weighting to emulate baseline randomizations based on age, sex, diabetes severity, comorbidities, sociodemographic profile. And we also used, when we had those propensity scores, weighted regressions, whether those were semi-parametric, Cox regressions, or non-parametric. And generally, we use constant hazard ratios as a measure of drug comparative effectiveness. But following recent literature from Cole and Hernan, we also quantified the risk of dementia onset and death without dementia using cumulative incidence functions, CIFs, and their representative curves, the cumulative incidence curves, or CICs. So why using a competing event framework? Though our primary outcome of interest is Alzheimer's disease, any patient is also at risk of death. And so here is a graphical summary of the 3 potential paths that one could consider. The patient could follow path 1 and develop Alzheimer's disease. A patient could also follow path 2 and die without dementia. And there's a third path, actually, that is follow path 1, develop AD, and then follow path 3 to death. And in this study, we're only considering the competition between path 1 and path 2. And so we ignore all the events conditioned on already being diagnosed with Alzheimer's disease. So we do not consider path 3 in this study. So the benefit of such a framework is twofold. First, we avoid misestimating the probability of event occurrence, so the probability of dementia onset occurrence, and the relative effects of certain covariates on outcome incidence. And second, it's also more meaningful clinically because physicians are decision makers who may need to consider different risks at a given point in time. So let's now go over some of the statistical components of this work, and we can discuss this during the Q&A session as well. So because we have a competing risk framework, 2 distinct hazard functions are involved, one for dementia onset and one for death without dementia. And in the main analysis, we considered 2 Cox proportional hazards models with metformin's treatment as the only covariate, because we had already adjusted for confounders in the propensity score model. And the proportionality factors are exponential of beta 1 and exponential of beta 2. And the baseline hazard functions are HO1 and HO2. And from there, we derive the causal estimates of interest in addition to the hazard ratios that are constant. The advantage is that the other causal estimate of interest are time varying, and that's more easily interpretable. So if we denote by T subscript T, the time to AD or death, whichever event occurs first, enter the anti-diabetic treatment regime big T, which is 0 for the control drug, sulfonylureas, and 1 for the drug of interest, metformin. And if we denote by E of T, the indicator for that first event, whether it's AD or death without AD, we can express 2 cumulative incidence functions, 2 CIFs, and 1 for each of the 2 outcomes that are already indexed by the target trial arm. And so we can also define them per drug. So we can either have 2 CIFs or 4 CIFs, depending on the framework we adopt for the modeling, whether it's fully, it's semi-parametric or non-parametric. And so both cumulative incidence functions involve this survival function, which is S of T, that represents the overall survival under treatment regime T, whether it's sulfonylureas or metformin. But they differ in terms of the product with the cause-specific baseline hazard and the hazard ratio inside the integral. So they have in common this S of T, and then they differ because of HO1, HO2, and the beta coefficients, beta 1 or beta 2. And if we try to express the survival function, so S of T, which captures the probability of no dementia onset and no death by time T, it reduces to the product of a function that only depends on our dementia setup parameters, HO1 and beta 1, with a function that only depends on the death setup parameters, which are HO2 and beta 2. However, these do not represent anything, any survival function in the context of the competing risk framework. But what's clear in that setup is that the risk of dementia onset at time T under treatment regime big T, whether it's metformin or sulfonylureas, depends through that survival function, through S of T, on the cause-specific baseline hazard of death. So that clearly makes the point of the competing event framework, the idea of not neglecting the risk of death without dementia at a given point in time. So here we do not use a doubly robust framework. And if we wanted to do so, what would change is that we would have additional terms that would enter the exponential here. So in addition to beta 1T or beta 2T, we would have the same covariates that we used in the propensity score model. So we would have, for example, age, sex, cancer history, etc. We can discuss these later. And the pH assumption, the proportional hazard assumption, can be relaxed. This is what we've done by defining 2 CIFs by outcome, 1 per target trial arm. So 1 for metformin, 1 for sulfonylureas. That way, we do not assume that there is this constant hazard ratio. And this results in a set of 4 cumulative incidence functions in a set of 4 cumulative incidence curves that are not linked with each other beyond the dependence that's introduced by S of t, but the overall survival function. And so that's easier also to interpret those counterfactual words without having the extra assumption of proportional hazards. So now to dive into the set of confounders that we've used in this study. So when trying to take a causal inference approach, uncovering those sources of confounding matters, and what we define as confounder is a variable that would typically weakly influence the treatment variable, but strongly influence at least one of the outcomes. That can be dementia, that can be death without dementia, or that can be both. And in our scenario, one can think of the following: sociodemographic characteristics, markers related to the exposure, diabetes, and other comorbidities, as well as spatiotemporal factors. Because in the US and the UK, we've seen that the history of the drugs of interest, metformin and sulfonylurea, was different. So I'd like to briefly discuss the rationale for each of those covariates. So age being the largest risk factor for dementia onset and death, it was included as a covariate. That seems reasonable. It is unknown whether sex affects anti-diabetic treatment assignment, but it strongly relates to the death outcome, and it may affect dementia onset outcome beyond its effect on survival. And there might be sex-based differences in disease detection as well, and in reporting in the medical records, often resulting in women having a higher incidence of dementia than men, as documented in the electronic health record. As for the socioeconomic status, it is associated with both dementia and death outcomes, so it seemed reasonable to include it. We incorporated body mass index at treatment initiation. It's a marker of obesity and a risk factor for dementia. And of importance in our context, it is known to affect life expectancy, hence the association with our time to death outcome. And when I said it's associated with dementia, I meant that it is linked with higher levels of inflammatory markers, which are involved in the Alzheimer's disease development process as well. We added hemoglobin A1C baseline levels in the propensity score model. It measures the average blood sugar levels over a period of about 3 months, and also this captures the severity of diabetes, which is the underlying exposure. And hypertension, cardiovascular disease, and stroke were added as well. These 3 comorbidities are associated with dementia, and that's the reason why they were considered. Cancer is associated with time to death. And for a patient to be categorized as having a history of cancer here at baseline, we required at least 2 instances of cancer ICD codes in the electronic health record. And the choice of 2 was based on previous literature, which suggested that accuracy was higher with 2 instances of ICD-910 codes for cancer instead of 1 only. And we also made sure with manual review of certain electronic health records that it did not include a history of cancer within the family, but not affecting the patient. And in the UK CPRD cohort, we also controlled for smoking status and for chronic obstructive pulmonary disease, COPD, as they are both associated with time to death. And one thing that's important to mention is that while the US RPDR cohorts mainly included patients living in the Boston area, the UK was more representative of patients nationally. And so to adjust for this geographical heterogeneity, the region of residence was incorporated as a categorical factor in the propensity score model for the UK CPRD cohorts. So there were 12 levels as well as the level of reference. And since the UK CPRD cohort was spanning a longer time period from 2000 to 2017, as compared to the US, which was 2007-2017, the calendar year of the first anti-diabetic prescription was added to control for temporal changes in prescribing patterns, as well as mortality trends and age-specific incidences of dementia. And given that the US cohort captures patients who initiated after the new guideline from the ADA, there were no specific concerns that were related to a potential shift in prescription patterns, as we discussed before. So that's the reason why we did not include that additional covariate in the US cohort. So we validated this actually by looking at the number of new prescriptions of each drug per year in each cohort, the US one, the UK one, with a rather stable mix in the US. And in contrast, a downward trend in terms of the use of sulfonylurea as the first line treatment in the UK. So that's the blue bars that you can see here. So if we look now at the characteristics of patients in both cohorts, the UK one and the US one, here is a brief summary of what we can notice. So in the US, metformin initiators were younger at baseline than their sulfonylurea counterparts. They were also more hypertensive, and they had fewer missing values in terms of both their baseline BMI, in terms of their baseline BMI than patients that were initiating on the control drug. And in the UK, we observed the same age pattern. Metformin patients were also more likely to have entered the cohort more recently. They had lower hemoglobin A1C at baseline, as well as higher BMI at baseline. And this makes sense because of sulfonylurea's tendency to promote weight gain, justifying their preferable prescription in the case of non-obese patients. And similar to the US, in the UK, they included more cardiovascular disease, more hypertension cases, but fewer cancer cases at baseline than their sulfonylurea counterparts. So the age pattern confirms actually a large-scale analysis that was conducted in 2018 on over 1 million patients in the US, which found mean age of 57 and 64 at treatment initiation for metformin versus sulfonylurea patients. So this pattern that we observed when comparing the two groups of drug takers was expected based on the previous literature. So while looking at these tabulated and sometimes boring statistics, well, one of the three assumptions that's needed to identify the average treatment effect, the ATE, is positivity, which means that across all levels of covariates that we consider, where at least one drug taker is represented, one metformin patient for one sulfonylurea patient, there should be samples of both metformin and sulfonylurea arms. So to check that assumption, I also considered the Cartesian product of all the possible characteristics to confirm the validity of the assumption in the US RPDR cohort. That involved discretizing the continuous age variable into 4 categories to make the verification more tractable. And in addition to positivity, the second identifiability criterion for ATE is conditional exchangeability, which means that given the propensity score, which we can assume is a summary of a patient characteristics, the counterfactual outcomes should be independent of treatment. And per the standard inverse propensity scheme, the lower a patient's propensity to receive the treatment they were given, the more weight they will have because they're more rare. And here, following guidance by Hernan and Robbins, we use stabilized weights, which just involves multiplying by the probability of being assigned metformin on the numerator. And this was done to reduce the range of individual patients' weights and limit the influence of potentially low or potentially high weights, so the outliers. So yeah, the only difference is this probability that t equal 1, the overall prevalence of the treatment of interest metformin in the study cohort, whether it's the UK one or the US one. So graphically, we look at what weighting does to the overlap of the two populations, the metformin population and the sulfonylurea population, with the idea of checking that when we emulate our trial, we're doing as if it was random. So here we look at the density of age, for example, baseline in the US cohort. Prior to weighting, there is an imbalance between sulfonylurea and metformin. As we've seen earlier, metformin patients tend to be younger. And after the weighting procedure, we achieve much better overlay, and the two density functions overlap better. So the same observation could be made in the UK with the same age pattern at baseline and the same improved overlap after the weighting procedure. And what we've seen with the age variable applies actually across the board to other confounding factors. And this plot shows the absolute standardized mean difference between metformin and sulfonylurea patients along different variables pre and post weighting being the red dots. The major differences between the two arms reduce after we apply the weighting procedure, which is was expected. And the same goes with the UK cohort, except that we have more covariates. And this is true except for one, which is hemoglobin A1C. As you can see, the red dot, which is post IPTW, is more on the right side than the dot prior to any weighting adjustment. And so that might happen, that has been investigated in a JASA paper, and reasons for that include a potential lack of overlap in covariates for certain patient subgroups. So in certain strata based on race, ethnicity, for example, that could be also based on sex, there could be differences, and that induces positivity issues, lack of overlap, and even with weighting that can lead to a reverse scenario as compared to what's expected. So let's now look at some of the results. Our first outcome of interest was time to death. We compared the effect of the two drugs on all-cause mortality in both cohorts. For this, we used a Cox proportional hazard regression model with IPTW to mimic this randomization at baseline. So metformin here significantly improved survival compared to the other baseline anti-diabetic treatment, which was sulfonylureas. In the US cohort, however, there was no evidence of heterogeneous treatment effects, either by age at treatment initiation or by sex or BMI at baseline. In the UK, we obtained the effect of metformin on all-cause mortality in the full population. However, the subgroup analysis revealed evidence of a stronger effect among patients initiating treatment at a younger age, before age 70, and among those with higher baseline BMI. So that's the type of trend you can see here in the forest plot, the one by age group and the one by BMI level at baseline. However, there was no difference based on sex, at least in the cohorts that we've studied. So we note that the survival curves between the two emulated trials, so here the blue one for metformin initiators and the pink one for sulfonylurea initiators, these two separate approximately 3 years after treatment initiation. And this trend actually persists for a long time until the end of follow-up, which was 12 years in the US and 16 years, as you can see here on the graph, for the UK. So as we discussed earlier, in addition to these hazard ratios, which are sometimes problematic parameters for causal inference, but are still traditionally used as effect measures in the medical literature, we also quantified the time-dependent, time-varying cumulative incidence functions, the CIFs, for each of the 2 outcomes of interest. So dementia onset in blue cues here, and death here in orange or red cues. And the CIFs here characterize the probability of event occurrence, so the probability of dementia onset occurrence or the probability of death without dementia by a certain time, which varies between 1, which is the end of our running time period, until you're 12 or until you're 16. And so what we call the average treatment effect, or the ATE, is the difference between pairs of CIFs. So the dementia onset CIFs or the death without dementia CIFs. And the advantage of these cumulative incidence curves is that they provide a visual overview of risk following drug exposure, following anti-diabetic drug initiation, which is more interpretable for clinical trial design and helps us make recommendations for improving the design of subsequent trials. So we chose the risk difference between those 2 curves as a summary of treatment effect, but there are other options. For example, risk ratios could be considered. So before I switch to the next slide, I'd like to highlight 2 observations. The blue colored curves, dementia onset in each treatment regime, were close to each other, while the orange colored curves visually separate. So that's really obvious when you look at the UK plots on the right of the slide. And second, the magnitude of the probability of dementia onset in blue was generally higher in the US on the left than in the UK on the right. So that will help us for the next slide, which showcases the risk difference of each cohort, the US and the UK. So overall, the benefits of metformin can be interpreted in terms of both delaying dementia onset and prolonging life without dementia, as represented on these 2 charts. Though clinically minimal, the dementia risk difference in the US cohorts taken as a whole consistently remained below 0, which is this dashed line here, serving as a reference. And so that means that metformin conferred a lower risk of onset in this population, with cognitive decline delayed by about 4 months if taking the drug for 12 years. So adherence is definitely, one limitation and an aspect that we should consider later using time-varying propensity score models. So the 5-year risk difference in the UK cohort was smaller, and it changed over time. So as you can see, this risk difference in blue in the UK crossed the 0 reference line at about 8 years, 8 out of 16, which was our maximum follow-up time. However, the risk difference for death without dementia, so the orange colored curve, was significant and clinically meaningful in both data sets. It was of larger magnitude in the UK, as you can see here. So the scale is the same on the 2 graphs. So you can see that if taking the drug for 16 years, that's about 15 months approximately of gain, life gain. So the risk difference curves for dementia onset and death without dementia were much closer, as you can see in the US cohort relative to the UK. And one reason for this could be the population structure difference. I'm just reading one question in the chat. Yeah, okay, like bias by indication. Yeah, we can discuss this during the Q&A. Yeah, so there are definitely differences in the population structure of metformin takers versus sulfonylurea takers. So the baseline age distributions in the US and the UK do, are not similar. In the US, as you can see, the mean and median age is much higher than in the UK. So that's one difference between the two cohorts. The UK CPRD also had a higher death rate than the US one. And this affects the total number of patients that are at risk over time. And the US rates of diagnosed dementia in both treatment arms, in both metformin, the metformin population and the sulfonylurea population, were higher than the death rate. So that's what you can see here. The blue curves, the blue cumulative hazard of dementia onset are above the death without dementia curves, and the reverse pattern can be observed in the UK. So this is a difference in the end-of-life outcome patterns that we observed between the US and the UK populations. So taken together, the baseline age pattern difference and the end-of-life outcome difference, these are population characteristics that affect the proportion of patients at risk over time in each target trial arm. And understanding differences between populations better is key to the design of future clinical trials, which should include a more geographically and more socio-demographically diverse set of patients. This is especially true for the US cohort. So I'd like to add that we conducted a sensitivity analysis to assess the robustness of our results. And for this, we used a non-parametric approach where there is no proportional hazard assumption involved. There is a model for each drug outcome pair in each database of interest. So relaxing the proportional hazards assumption led to consistent results here in terms of dementia onset and death without dementia outcomes, as showcased here on the graph by the overlay between the smooth curve, which is the Cox proportional hazard obtained curve, and the more noisy or wavy curve, which is the one obtained under the non-parametric framework. And the same was true of the dementia onset outcome in the UK, which is the blue curve here. However, approximately 8 years after the beginning of the emulated trial, the two risk difference estimates for the death outcome deviate from each other. So on the right plot, you can see this deviation between the smooth curve again and the noisy wavy curve. That is for the death without dementia outcome. However, for dementia onsets, there was no problem when relaxing that assumption. But to us, that is something that should systematically be checked for, whether it's a randomized trial or an emulated trial, whether there is sensitivity of the results, especially when we have long periods of follow-up, depending on the assumed model structure. So the role of this is to validate graphically the proportional hazard assumption under which the hazard ratio is constant over time rather than time varying. And here, again, there's good alignment between the two versions of the hazard ratio for each outcome in the two cohorts, except here, as we noticed earlier, the horizontal line and the curve, which represents the time varying ratio, the time varying beta coefficient. So yeah, except for the UK cohort, for that death without dementia outcome, there's generally good alignment between the constant beta and the beta of t, which doesn't require the proportional hazards assumption. And since age is the principal risk factor for dementia, what we did is that we further investigated the possible effect modification by age group at treatment initiation. So we mentioned this earlier, but we stratified based on that threshold age of 70. And did the same procedure again, but within each subgroup, the group of patients who got prescribed early on and the group of patients who got prescribed for the first time later. And we also stratified the original population by sex and by baseline BMI level, very similar to the cause mortality analysis that we did earlier. And the reported hazard ratios here correspond to cause-specific Cox models with dementia onset as the outcome of interest. And there's one model for each stratified analysis. And the beneficial effects of metformin on dementia onset was highest in patients who started anti-diabetic treatment earlier, before age 70. That can be seen here by the pattern, especially in the US cohorts. The difference was more pronounced there. And this is perhaps clearer when looking at the comparison of risk differences over time between the two cohorts. Although there were fewer patients who started treatment after the age of 70, and the older stratum, of course, contributed less person years, this age-specific finding suggests that the drug may be especially beneficial among patients who initiate anti-diabetic treatment early on. And here, yeah, there is overlap, and it's consistently, below zero for the dementia onset and death outcomes. And here in the UK, it's clear that there is a differential effect when we talk about the dementia onset outcome or the death without dementia outcome. And none of the other measured factors modified the effect of treatment, of metformin treatment, in the two patient populations we studied. So the main modifying aspect was due to age here at treatment initiation. And such stratified analysis here with very basic characteristics, age, sex, BMI level at baseline, hemoglobin A1C level at baseline, can inform the planning of future clinical trials in that it explores the source of the signal in subpopulations defined by factors that are rather simple, right? Factors that can readily be implemented as an inclusion-exclusion criterion in a randomized clinical trial. That could be, for example, age enrollment in the trial. And we conducted a sensitivity analysis to that threshold value. I told you we considered 70, but we also looked at the results obtained, with a cutoff at 65 or 75, just to make sure that this is not an arbitrary cutoff and that the results are consistent. So finally, and I'd like to end on that, we also explored whether the drug of interest, metformin, acted primarily by a better control of blood sugar, as measured over time by the hemoglobin A1C levels. In addition to a direct path from treatment metformin to outcomes, dementia onset and death without dementia, one could imagine this intermediary path. So one could imagine that there's mediation that involves diabetes management, so that is through a better control of hemoglobin A1C levels. And we could adopt a causal mediation framework. That's something I'm planning to do later, to measure this indirect effect via the control of hyperglycemia. But given that it was a complementary analysis for us, what we did is that we considered a linear mixed effect model for hemoglobin A1C repeated measures as a first approximation of this. And given the lack of structure in longitudinal lab measurements in the UK CPRD database, that was not possible for that cohort, but we did so in the US cohort at least. And there, 77% of patients had at least one hemoglobin A1C measurement post-baseline, so that we could consider this repeated measurement framework. And we applied a repeated measures model on all hemoglobin A1C values after the 3 months treatment mark. So this is on average the time it takes for metformin to start acting on a patient's diabetes. And doing this, we found that although the average level of hemoglobin A1C was significantly lower in metformin initiators, the effect size, which is between -30 and -15 percentage points, is clinically negligible. So that suggests that if any effect of metformin on the risk of dementia, it is likely through mechanisms that are that differ from the control of blood sugar. And this is what motivated the team, the systems pharmacology branch of the team, to conduct further investigations. I could talk briefly about this if this is of interest. This is not the work I've done, but I've contributed to the design of those studies. And so it would be nice to have your feedback on how we can improve the experimental part of this study to better characterize the actions of metformin on human neurons, because it has not been characterized extensively. So just to summarize what we've done, it's treating differentiated human neural cells in culture with metformin and glyburide. Glyburide is a specific drug from the sulfonylurea drug class. We could do the same with other drugs. And that was done at biologically relevant concentrations that approximate actual concentrations in the cerebrospinal fluid and in plasma. And two timeframes were considered, 24 hours and 72 hours. And yeah, after RNA sequencing, 100 differentially expressed genes that were significantly altered in a dose-dependent manner. So for 10 and 40 micromolar were identified. And then that can be related to the different pathways that are involved. And so that helps us identify candidate biomarkers. And in summary, one of them, that was validated using other approaches, including RNA levels in the human brain, was SPP1 or osteopontin. So that's one protein that could be used to track cognitive impairment, especially among asymptomatic patients. So that would be like a bit invasive to track the levels of SPP1, but that's something doable and something that the part of the team that's involved in small-scale clinical trials at Mass General Brigham is currently doing. Yeah, so I mean, I could share my slides later for next steps, just to have your feedback. And I'm happy to take any questions, especially one question on the chat was about the limitations, especially confounding by indication, the potential residual confoundings, etc.
Merci Marie-Laure. Donc on a le temps pour quelques questions. Est-ce qu'il y a des questions? Pour l'instant, pas forcément. Donc je vais en profiter pour. Hélène, vas-y, tu lèves la main, tu peux poser ta question.
Merci. In English or in French?
Oui, n'importe. En français, c'est possible.
Je voulais savoir si vous aviez fait des analyses de sensibilité concernant le modèle pour le propensity score. J'ai vu que vous aviez posé un peu la question de A1, mais par exemple, l'éducation, vous ne l'avez pas du tout, c'est pas possible de l'utiliser.
Ouais, non, enfin, on pourrait faire à la fois au niveau des covariables utilisées. Donc, ça, on a un peu fait. Au début, par exemple, on ne prenait pas en compte l'historique de cancer et après, on l'a inclus comme variable de contrôle dans le modèle de propension. Et on pourrait, ça, c'est un truc que moi, je voulais faire plus tard. Ce n'est pas vraiment d'intérêt pour l'équipe, mais je voulais tester, par exemple, des forêts aléatoires. Parce que ça permettrait de mieux comprendre les interactions entre les différentes variables. Et surtout, après, avec les forêts aléatoires, on peut essayer de mesurer les différences des variables de sortie dans chaque feuille, dans chaque sous-branche. Donc, c'est plus interprétable. Et moi, je voulais tester surtout si ça améliorait la balance, l'équilibre entre les deux bras. Parce qu'au final, que ce soit une régression logistique ou une forêt aléatoire pour la classification ou des forêts, mais avec du boosting aussi. Ce qui importe, c'est juste qu'on obtienne la meilleure balance entre les bras. Donc, comme je sais qu'il y a des éléments à améliorer, par exemple pour les niveaux d'hémoglobine glyquée au départ, moi, c'est un truc que je veux continuer à poursuivre. Mais en revanche, en ce qui concerne l'éducation, ou par exemple, est-ce qu'il y a un régime, une diététique particulière qui a été suivie, ça, c'est des éléments, du coup, de management du diabète, de contrôle du diabète qu'on n'a pas, et que ce soit dans les données structurées de l'Angleterre ou les données du Royaume-Uni ou les données structurées des US. Par contre, un truc qu'on peut faire et qu'on a commencé à faire, c'est d'essayer de trouver des informations dans les notes cliniques. Par exemple, quand le niveau d'entrée de l'hémoglobine glyquée ou quand l'indice de masse corporelle n'est pas indiqué dans les données structurées, on a vu en faisant soit des regular expressions, juste en cherchant HB ou d'autres mesures qu'on peut retirer de l'information et donc du coup limiter les données manquantes. Donc ça, c'est quelque chose qu'on a commencé à faire. C'est plus facile à faire pour nous dans les données, dans les notes cliniques des US, parce que les notes cliniques de l'Angleterre, tout le monde dans l'équipe n'y a pas accès. Donc voilà, mais au moins sur tester ça, si on inclut une note clinique et qu'on limite les données manquantes, alors en quoi ça change la balance qu'on obtient avec la régression logistique versus des forêts aléatoires ou des modèles qui ont du boosting. Donc voilà.
D'accord, merci.
Ok, et du coup, juste rapidement, avant qu'on n'ait plus le temps, il y a Rodolphe qui demandait, il posait une question sur le fait qu'il y avait toujours un biais d'indication qui semblait persister avec la sulfonyle par rapport à l'autre, à la metformine. Tu peux commenter là-dessus?
Oui, mais un truc qu'on voulait faire parce que, comment dire, pour l'instant, c'est quelque chose sur lequel on travaille. On n'a pas de méthode encore systématique pour estimer les préférences de chaque médecin. Mais il semble qu'à travers les 8 centres qu'on a aux US, notamment il y en a dans des cliniques assez rurales, par exemple New Hampshire et Vermont. Nous, ce qu'on voudrait faire pour, essayer de se débarrasser, on va dire, de ce biais, c'est utiliser comme instrument la prescription, la préférence de prescription du PCP, du médecin traitant, et du coup voir dans un premier temps en quoi cette préférence de prescription elle conditionne, elle influe le traitement avec tel antidiabétique. Donc après, bien sûr, on peut toujours contrôler si on veut avec les variables d'ajustement, l'âge, le niveau, par exemple, des problèmes rénaux, etc. Et ensuite, faire un second ordre, du coup, pour nos 2 sorties d'intérêt, qui sont le temps jusqu'à l'incidence de la maladie d'Alzheimer, de la démence, ou le décès, mais sans que la démence soit impliquée. Donc, du coup, ça pourrait être une technique. Donc, je dirais qu'une technique, c'est déjà d'essayer de limiter pour nous les données manquantes pour ne pas avoir ces covariables qu'on n'aime pas trop, juste de 1 et de 0. Après, on peut bien sûr toujours imputer, etc. Après, comme on disait tout à l'heure, essayer de compléter avec les notes cliniques, mais une autre, en fait, quand on ne peut pas avoir accès à trop de covariables sur le profil du patient, c'est d'utiliser une autre technique d'inférence. Et pour l'instant, ça nous semble assez approprié d'essayer d'utiliser cet instrument-là. Et la raison, elle est assez simple, c'est que, en fait, chaque patient, du coup, est associé à son médecin traitant, et que ce soit au UK ou aux US, on peut on peut juste quantifier pour tous les patients de ce médecin quelle est la préférence de traitement. Et on pourrait aussi, soit on peut prendre juste sulfonylureas et metformin, ou on peut prendre aussi tous les autres antidiabétiques qui prescrivent. Donc, le dénominateur, c'est un peu notre choix. Il faut tester si on prend en compte tout, si on prend en compte juste les deux auxquels on s'intéresse. Mais du coup, on peut définir un pourcentage annuel de prescription, nouvelle prescription de tel médicament versus tel autre. Et après, on peut voir si on a envie de le faire overall ou juste sur la cohorte de patients qui vérifie tous les autres critères d'inclusion, exclusion. Et donc, voilà, l'avantage, c'est que c'est une variable qu'on peut rendre continue. Et du coup, on peut soit jouer avec un instrument continu, soit jouer un instrument binaire en mettant un seuil de préférence. Donc, ça nous semble être la technique pour l'instant la plus appropriée pour essayer de se débarrasser du biais d'indication. Mais si vous avez d'autres idées...
Mais je vais laisser, moi pas particulièrement, mais je vais laisser la discussion se continuer. Et si jamais tu peux en reparler avec Rodolphe à l'occasion ou avec d'autres, vous pouvez échanger. Parce que là, on a déjà dépassé le temps, donc on va s'arrêter là. Merci encore Marie-Laure pour ta présentation. Et le dernier séminaire de l'année, c'est la semaine prochaine avec Benjamin Hibert qui présentera son travail. Donc merci à tous pour votre participation et puis bonne fin de journée, à la semaine prochaine.